Okay, now we're getting to the fun part. So, DNA replication. DNA replication is a process of making more DNA or copying DNA. So, how do we copy the DNA? Well, first, we actually take an existing DNA strand and we separate it. We separate an existing DNA molecule. We pull apart the two strands of the molecule. So remember, a DNA molecule has two strands side by side. This would be one strand here, this is the other strand. And they're held together by these hydrogen bonds. So the first thing we're going to do is break those hydrogen bonds and separate this DNA molecule. Then we're going to use the help of some enzymes. And we're actually going to use the code from the existing strands, from the old strands. We're going to use their code to make a new strand. The new strand will not be the same as the template strand. It will be complementary. So what does that mean? Well, let's take a look. Show you a picture. So this is our original parental molecule of DNA. Well, if we pull this apart, the left strand says ACGAT, ACGAT, so nothing's changed with the parental strand. The right strand was TGCTA. It's still the same thing. Nothing changed with our right strand. But now we're going to use these blue parental strands as a template for building new complete DNA molecules. Remember, every nitrogenous base can only pair with one other possible base. T always pairs with A. G always pairs with C. So when we pull these apart, we can basically fill in what we're missing here. And we know what we're missing because there's only one possible option. The only thing that we could bind right here would be an A. The only thing we could bind here would be a C. The only thing here is a G. The only thing here is an A. The only thing here is a T. So we can fill in what we're missing by using the base pairing rule. And when we're all done, we're going to end up with two identical daughter DNA molecules. They are exactly the same as the original parental molecule. In fact, half of the new molecule is actually made from the previous parental DNA molecule. So every time DNA is copied, half of the molecule is old and half of the molecule is new. And both of these DNA molecules are identical. So this is called the semi-conservative model of DNA replication. And to conserve something means to save something. So semi-conservative means we're partially conserving the original model. The original parental DNA molecule, we are semi-conserving this molecule because actually when we finish, we still have part of the parental molecule here, exactly half. Each new DNA helix has one old strand and one new strand. This picture shows a little bit more realistic view of what DNA looks like. But what we're going to do again is pull apart and separate these hydrogen bonds. We're going to pull apart these old strands, and then we're going to fill in what we're missing here. So we pull them apart, and now I want you to think about it. What do you think would go right here next to the A? Could we fill in this missing nucleotide? What would go next to the C? What would go next to the G? And so that's how this process works. We pull apart the parental molecule, and then we use nucleotides that are floating around in the nucleus. We use nucleotides to fill in what we're missing to build this new strand, one nucleotide at a time. Well, I know that we needed a T here. We need a G here. We need a C here. Okay, same thing on this side. Well, here it looks like we need an A, and then we need a C, and then we need a G. So we're going to build this new strand by just adding in one nucleotide at a time for whatever we're missing. The end result is two identical DNA molecules. They look exactly like the parental molecule.